Hi guys, my name is Sridevi. I am pursuing BTech third year from Department of Biotechnology as a part of assignment Genetic Engineering. Now I am going to discuss about gene editing. You know what is gene editing? Gene editing or genome engineering is a type of genetic engineering in which DNA is inserted, deleted, or modified or replaced in the genome of a living organism. Genome. A genome is the genetic material of an organism. It consists of the DNA, RNA. The genome includes both genes, the non-coding DNA, and the genetic material of the mitochondria and chloroplasts. Now, what is targeted gene modification? The targeted gene modification via chimeric genome editing tools. There are three tools are used for genome editing, that is ZFNS, gene fingerprints, and TALENS and CRISPR-Cas9 system. Is the powerful methods to access a gene function and precisely manipulate cellular behavior and function. Genome editing, this approach is called as reverse genetics. Among the key requirements of the reverse genetic analysis is the ability to modify the DNA sequence of the target organism. Genome editing is of two types, natural methods and engineering methods. There are three methods that were used for plant genome manipulation. They are classical breeding, targeted genome editing, and transgenic approach. And these are the three modifications are done. That one is conventional breeding, gene modification, gene editing. You know what is conventional breeding? Conventional breeding is a selective breeding methodology where crops are selected based on superior performance. Pure line selection, small selection, backcross breeding, recurrent selection, hybridization were most famous traditional breeding methods. It is a longer breeding method and over dependent on phenotype and plants. You know what the gene modification and genome editing. Then the comparison between traditional and modern genome editing technologies. Now the difference how the gene modifications are done in traditional way and now the engineering technologies are modified. This also table is given clear explanation, and also the gene editing has advantages and disadvantages that are physically and biologically. Now why have to gene editing? Because of to understand the function of a gene or a protein, one interfaces with it in a sequence specific way and monitors its effects of the organism. In some organisms, it is difficult or impossible to perform spike-edit mutagens, and therefore more indirect methods must be used, such as the slicing is the gene of interest by short RNA interference, and also some times a gene disruption by sRNA can be variable or incomplete methods. And here some of the requirements are used for genome editing or genetic home devising. Homing devising, it is used for specific identification of the target sequence. And then endonuclease, it is used for creating double stranded break. And some of the uses for genome editing, that was the you have to target a gene or the specific mutation, promoter study, that's all. And here some of the tools for genome editing, that was meganuclease, zinc finger nuclease, TALENs, and CRISPR-Cas9. The CRISPR transfer clustered regularly interspatial short palindromic repeats. The palindromic, what is palindrome? It is a inverted repeat sequence. Further, the first tool of genome editing, that was meganuclease. It is first tool for used for double stranded break include gene manipulation. These engines binding state and restriction sites occur within same unit, hence difficult to modify. Here also some limitations are there. Example, we have to see mice, trait it be herbicide resistance and reference. And the second tool is zinc finger nuclease. It is a hybrid restriction enzymes. It consists of two parts that were generated by fusing a zinc finger DNA binding domain into a DNA cleavage domain. Here the structure of zinc finger nuclease, we have to observe clearly, we know the function, how does it works also. And here the mode of action of genome editing. The basic steps for genome editing is that binding of ZFN to DNA, restrictions the DNA and cut the sequence. After that break, these are the basic steps. And the third most of the system, that is a CRISPR-Cas system. These are the part of the bacterial immune system which it detects and recognizes the foreign DNA and cleaves it. The CRISPR is a traditional modern technique nowadays used to genome editing. And the components of CRISPR: protospacer adjacent motif and CRISPR RNA transactivating cRNA. You know, can genome editing be useful in eukaryotic cells? Yes. Based on engineered or bacterial nucleases, the development of genome editing technologies has opened up the possibility of directly targeting and modifying genome sequences in almost all eukaryotic cells. And here there's some different Cas proteins and their function. How does the Cas protein works? And example, we have to know the how the process happen. Here we have to see type 1, type 2, that the process is Besar equation, and like this. And here the structure of cRDNA and some of the action performed by CRISPR in bacteria. The CRISPR immune system works to protect bacteria from repeated viral attack via three basic steps. That one is adaptation and production of cRNA and targeting. Let's comes to how the mechanism works in genome editing, and these are the total content of the gene modifications and gene editing. These are the types and ingredients, all this. And some of the applications are there in agriculture. We are using gene editing. It can be used to create high degree of genetic variability at precise locus in the genome of the crop plants. It can be used to eradicate unwanted species like herbicide resistant weeds, insect pests. So many applications are there for using of agriculture to genome editing. And here the final conclusion is there. The advantage of genome editing is to hold the potential to cure genetic disease and create crops resistance to drought. But scientists need to work closely with the law and policy makers to ensure the technology can be used for benefit of mankind while minimizing the risk. What could gene editing do? The benefits of gene editing to humanity could be significant. For example, making a single base change in people's DNA could be a future treatment for a sickle cell disease or genetic blood diseases. So many diseases are worked out with the gene editing. And we have to must know how does gene editing work. Gene editing is a technology that can change DNA sequences one at more points in a strand. Scientists can remove or change a single base or insert a new gene altogether in. These are the technology. There are different ways to edit genes, but the most popular technique nowadays is more accurately we are using the technology is CRISPR-Cas9. It is assisted by a strand of RNA molecule similar to DNA. Depending upon the situation, what the scientist wants to achieve, they can just remove a segment of the DNA, introduce a single base chain, and repair a process, take over a clue that cuts the back together. And this is all about the gene editing, gene modifications in the organism. Thank you.